Hey everybody, Mr. Grove here. Today we're going to talk about bacteria. So bacteria, unlike viruses, are living, and they have, they meet the criteria of living things. They belong to the domain bacteria and then the kingdom bacteria as well. And so how are they different from you? Well, you're a eukaryote, whereas they are domain bacteria. And so they are no nucleus and no organelles. So unlike a eukaryotic cell that has all kinds of good stuff like the Golgi apparatus and mitochondria and things like that, they do not have any membrane-bound organelles. They're very small, but they're not so small that we can't see them. We can see bacteria with just standard microscopes very easily, unlike viruses that require electron microscopes because they are so small. Bacteria are also going to be single-celled organism, unlike you. You are a very multicellular organism made of trillions and trillions and trillions of cells. Chromosome-wise, bacteria typically just have kind of one major chromosome, unlike you that has 46 chromosomes. But they also kind of have this little circular DNA. They're very good at uptaking foreign DNA in the form of plasmids, and so that little circular piece is what we would call a plasmid. And we utilize that a lot in our biotechnology techniques. They reproduce asexually and a little bit sexually as well too, but asexual is definitely the primary way. That kind of has a special name for bacteria called binary fission. So it's basically kind of like mitosis. They have one cell that splits into two, and those two cells are genetic copies of each other. Some bacteria do have flagella. And then some bacteria thrive in aerobic environments, and then some are more anaerobic. The presence of oxygen is a negative to them. So a lot of times with like a burn victim that's severe burn, we'll put them in an oxygen tent. And that's not because they can't breathe very well. It's because they're at high risk of a bacterial infection because their skin's been damaged. And your skin is kind of your first line of defense. And so that is kind of some basic things about bacteria cells. Anatomy-wise, so we know that they have the nucleoid region, like we said, that's where their genetic information is. They have little plasmids that they can bring in foreign DNA. They also have ribosomes, so unlike viruses, they can make their own proteins. And then they have these layers, so the outside is going to be the capsule, then we have the cell wall, and then the cell membrane. And so the capsule is going to provide a little sticky coat and it works along with the pili to help transfer bacteria from that uninviting place to a real party-like environment for them. The cell wall gives a little bit of structure and rigidity, and then of course the cell membrane will allow some things in and some things out of the cell. So that let's talk about that party-like environment. There's really basically three things that we need for bacteria to really thrive and therefore reproduce. And that is warmth, and then we want moisture, and then we want organic material. So if we've got those three things, a bacteria can really do well. So warmth, the ideal is 37 degrees Celsius. Which is body temperature. If you notice, hospitals are usually kind of cold. Well, part of that's by design. If we lower the temperature just a little bit, that might keep bacteria growth inhibited. Moisture, so they got to have moisture to be able to grow. So a real cold, dry surface, not ideal for bacteria. And then there's got to be organic material. They do feed on organic materials, and so that is a necessity for a good quality bacteria growth. And so by removing those things, then we can kind of inhibit that. Let's talk some shapes. Okay, so shapes are very important in helping us kind of group and classify bacteria. So the three basic shapes are rods, spheres, and spirals. The official names of those would be bacillus, coccus, and spirillum. So spirillum and spiral, that should be pretty easy to remember. So just kind of remember rods and bacillus, spheres and coccus. And so that gives the general shape of the cell. Then we can add a prefix on, so we can either call it strepto or staphylo. So if it's a staph, it's going to kind of cluster up like grapes. And then if it's a rod, it's going to kind of link, or I'm sorry, if it's a strepto type bacteria, it's going to link together in like a chain. And so if we have like a streptobacillus bacteria, then that means we would have rods that are linked together. If we have a staphylococcus bacteria, we would have spheres that are clustered together. If we have a staphylobacillus, we would have rods that are clustered together. So this gives us even more ability to kind of group them into various groups and help us separate the good from the bad types of bacteria, because not all bacteria are harmful. There are a lot of good bacteria out there as well, and some that we absolutely need to be healthy. Another way to do that is through gram staining. So we have what's called gram positive and gram negative. So through a series of stains, that will allow us to end up with kind of a purplish colored bacteria or a pinkish colored bacteria. If it's purple, that means it's positive. And antibiotics typically work well against some kind of classic penicillin family antibiotics. One of the reasons being the cell wall has a thick layer of peptidoglycan, and those antibiotics can kind of penetrate the cell wall, and that can cause the cell then not to function or work. If it's a gram-negative bacteria, that has a tendency to have a thin layer of peptidoglycan with an outer membrane that helps protect and shield that. And so the antibiotics can't get into the cell wall as well, and it's hard for them to be able to, impact that gram-negative bacteria negatively then that way. So gram stain is a technique that just helps us determine that. So now we know the shape, we know kind of how they're clustered together, and we can kind of determine gram positive and negative and really start to kind of hone in on what type of bacteria we have. As far as people in the line of bacteria, the main man is this guy, Alexander Fleming. And so Fleming is noted as the discoverer of the first antibiotic, penicillin. And so as the story goes, he kind of was a little bit sloppy. I can't judge myself as far as his lab was concerned. And he went off to vacation with some bacteria cultures that he was studying, Staphylococcus aureus. And when he came back, some of the cultures were dead. And there was some fungus around, probably some sort of mold. And rather than just throwing them out, he decided, hey, let's see, why did these things die? And so as he honed in on that, he realized, wow, these things are actually inhibiting the growth of the bacteria. And so he pursued that, and that led him to the first discovery of penicillin. And so this was a huge game changer in the aspect of the modern world. Prior to that, amputations were very common, and people would die from common infections. But after this discovery, right in time for World War II also, Alexander Fleming was, it was a huge contribution to mankind and society. So that's Alexander Fleming, kind of our first guy with the penicillin family antibiotics. One last thing here, and we'll talk about this guy, and this is the Alton giant, Robert Wallow. And so this is kind of close to where our school is, about an hour away, Alton. And he is the tallest man in recorded history. 8'11" and change. He had a pituitary issue that they did not want to risk surgery on. And so he grew, and you can see his stats were just staggering. If you've not seen a video, it's hard to wrap your mind around how tall he actually was. And his weight was 439 pounds at his death, too. So by no means would you consider him an obese man, but there was a lot of him there. And so that's a lot of mass in the form of 439 pounds. He had trouble walking towards the end of his life. He had to walk with a cane. He had that real heavy metal braces that were common in that day. And he had just gotten them adjusted and they were going out on a tour. He toured with like a shoe company as kind of advertising, and he size 37 shoe, by the way, and so he the brace was kind of misfitted and it rubbed against his leg, his ankle, and he got an infection from that, and basically he died from that very quickly after that. They tried some blood transfusions and surgery, but this was pre-antibiotic, and so because of that, a common bacteria infection, was what did him in. So the Alton giant, Robert Wallow, ended up dying from a bacteria infection. So hopefully that will give you a little bit of a mindset on how bacteria work commonly, and we'll kind of talk some more specifics about specific bacteria a little bit later. So hope that was helpful.